Strip away the enzymes, the proteins, and the nucleus itself, and the human genome reveals its identity as a physical polymer. This string of atoms remains bound by the rigid, inflexible laws of chemistry. Observe a eukaryotic replication fork inside a dividing cell. Then look at a thermal cycler executing a polymerase chain reaction in a forensic laboratory. One occurs in the aqueous, highly regulated environment of a living nucleus. The other happens in a sterile plastic tube. Yet both processes rely on the exact same physical forces to pull strands apart, bind new molecules, and synthesize data. Modern biotechnology does not invent new biological mechanics. Every synthetic achievement in genetic engineering is simply a laboratory isolating and exploiting the natural chemical limits of the DNA molecule. To understand how we manipulate life at the molecular level, we have to look at 3 immutable rules governing this polymer. 1. Thermodynamic pairing. 2. Electrostatic packaging. And 3. Backbone polarity. Abandon the idea of biology as an organic mystery. When you view the genome strictly as applied chemistry, the mechanics of advanced molecular genetics become entirely predictable. The central architecture of DNA relies on complementary base pairing. The 2 strands of the double helix are not fused together by strong permanent covalent bonds, but rather by non-covalent hydrogen bonds. This creates a specific thermodynamic threshold. Adenine pairs with thymine using 2 hydrogen bonds, and guanine pairs with cytosine using 3. These aromatic heterocycles follow Huckel's rule. Delocalized pi electrons travel through the rings, creating an exceptionally stable, unreactive molecule perfectly suited for long-term data storage. Capturing this physical structure was a matter of crystallography. Rosalind Franklin bombarded DNA with X-rays, producing diffraction patterns that revealed the exact spatial dimensions of this aromatic stability. Those hard physical measurements provided the foundation for James Watson and Francis Crick. By mapping the molecular dimensions, they deduced how the anti-parallel strands paired together, revealing the physical mechanism for copying genetic data. Because the rungs of the ladder are only held together by hydrogen bonds, the molecule can undergo denaturation. Subject the helix to enough environmental stress, like extreme pH or heat, and the strands will melt apart, while the covalent phosphodiester backbone remains completely intact. Inside a cell, biological replication relies on an enzyme called helicase. It acts as a wedge, physically forcing the hydrogen bonds apart so the genome can be copied. Polymerase chain reaction, or PCR, eliminates the need for helicase entirely. Instead of relying on a complex enzyme to pry the strands apart, the laboratory process simply cooks the DNA. The temperature is spiked until the hydrogen bonds reach their thermal breaking point and snap. There is a mechanical flaw with this method. The heat required to melt DNA also destroys standard human polymerase enzymes. To solve this, scientists turned to Thermus aquaticus, a bacterium evolved to survive in boiling hot springs. Extracting the polymerase from this extreme environment gave engineers an enzyme that operates flawlessly at near boiling temperatures. PCR is a brute force thermal exploitation of DNA's natural hydrogen bond limit. A human nucleus is roughly 6 micrometers wide. Into that space, the cell must pack 6 billion bases of DNA without shearing the polymer. The biological solution is extreme compression. The cell condenses the genome by wrapping it around octamers of histone proteins, creating repeating units known as nucleosomes. This packaging is driven entirely by electrostatic attraction. The alternating sugar-phosphate groups in the DNA backbone carry a heavy negative charge. Histone proteins, conversely, are uniquely rich in basic amino acids like lysine and arginine. At normal physiological pH, these amino acids carry a strong positive charge. Negative binds to positive. The severe electrostatic attraction locks the DNA tightly to the proteins, creating dense, highly compacted complexes called heterochromatin, where genes are transcriptionally silent. This charge dynamic creates a significant clinical hurdle. The chemotherapy drug cisplatin is designed to treat cancer by irreversibly binding to DNA, which blocks repair complexes and triggers cell death, but cancer cells quickly develop resistance. If the target DNA is buried deep within tightly packed heterochromatin, the cisplatin molecule physically cannot access the genome to bind to it. Data shows cisplatin alone fails to increase lifespan. The drug is locked out. But combining butyrate with cisplatin drastically extends survival. Why? Butyrate causes a massive surge in histone acetylation. Acetyl groups attach to histones, physically masking their positive charge. Without that strong positive charge anchoring it, the electrostatic attraction vanishes. The histones release the negatively charged DNA, unwinding it from dense heterochromatin into loose, exposed euchromatin. The chemotherapy drug can now reach its target. In this clinical model, overcoming drug resistance depends on manipulating basic electrostatic attraction to physically expose the polymer. The final immutable rule governing this polymer is the strict unidirectional polarity of its backbone. DNA has absolute structural asymmetry, a 5' phosphate group and a 3' hydroxyl group. This dictates enzymatic activity. Polymerases synthesize exclusively 5' to 3'. Because the helix is antiparallel, this creates a logistical bottleneck at the replication fork. On the lagging strand, the polymerase moves backwards. Biology's workaround is to synthesize the lagging strand in disjointed stutters, known as Okazaki fragments. This requires laying down constant RNA primers, building short fragments backwards, and finally sealing the gaps with an enzyme called DNA ligase. The polymerase stutters because of a core chemical dependency. To attach the next incoming nucleotide, the enzyme must have an existing 3' hydroxyl group to form the phosphodiester bond. Without that specific oxygen atom, synthesis stops completely. That single missing atom provides the exact vulnerability required for Sanger sequencing. In the lab, scientists synthesize modified nucleotides called dideoxyribonucleotides, or DDNTPs. A DDNTP is engineered with a highly specific structural sabotage. It completely lacks that critical 3' hydroxyl group. By flooding a replication cycle with a low concentration of these modified bases, engineers intentionally crash the polymerases. Every time a DDNTP is incorporated, the chain terminates. This yields millions of staggered fragments of varying lengths, which are then separated by size in a gel to decode the exact sequence of the bases. The Human Genome Project mapped 3 billion base pairs of DNA. That massive data extraction was achieved by weaponizing a polymerase enzyme's strict chemical dependency on a single oxygen atom. When we combine these rules, thermodynamic melting, electrostatic exposure, and polarity limits, we arrive at recombinant DNA technology, the process of splicing genetic code from entirely different species into a single functional sequence. The primary tool for this splicing is the restriction endonuclease. These enzymes evolved naturally in bacteria as a cellular immune defense, designed to lock onto and shred the DNA of invading viruses. These enzymes scan for palindromic sequences, where the opposing strands are identical. The enzyme executes a staggered cut, leaving unpaired bases exposed as sticky ends. Driven to form hydrogen bonds, engineers use these ends to drop a human gene into a bacterial plasmid. Once sealed by DNA ligase, the bacteria will read the foreign DNA as its own. This exact recombinant process is what allows the mass industrial production of human insulin and forms the basis for modern viral vector gene therapies. Whether a cell is deploying mismatch repair enzymes during the G2 phase of division, or an engineer is constructing a transgenic mouse model in a lab, the biochemical grammar remains perfectly identical. The physical vulnerabilities of the DNA polymer, its exact melting points, its susceptibility to electrostatic charge, and its strict directional limits serve as the specific features that make life adaptable and programmable. The perceived magic of biology is an illusion. True mastery over life is achieved only through a rigorous first principles understanding of its immutable biochemical rules.